What's up, guys? How you all doing tonight? All right, tonight, guys, I'm gonna be running a video where a doctor gonna be talking about how that herbal medicine and where it come from, and he basically gonna show you, give you an idea of how medicine has created. So just hang in there, guys, and let's listen to the video and see if we could gain something from it. This is not, I don't have the rights to this video, but I'm just running the video for informational purpose. As always, please remember to do your own research. This is informational purpose. Before you do use any product, please speak to your doctor before using anything mentioned in this video. That's a disclaimer. Speak to your doctor first. So this doctor is from Barbados, and basically he's gonna be talking about medicine, from the bush that they get in Barbados. He's gonna even show you how he get the medicine from the plant. I mean, it's a video about 45 to almost 50 minutes, but it's interesting. There's a lot of information to actually intake and learn from. So let's go, guys. I'm going to be playing this video, guys. So hold on.
Welcome and thank you for joining us on this series, Bush Tea, Herbs, Plants, and Botanical Stories of Barbados. I'm Aisha Gibson Gill, and this is a production of the National Cultural Foundation. Welcome back to our program. After discovering several amazing herbs and plants at Ngozi Farm and Sanctuary, we're meeting with Dr. Damien Kohal at the UWI to explore the drug pipeline as we look at the process of moving from a plant to a pharmaceutical.
My name is Damien Kohal. I'm a pharmacologist in the Faculty of Medical Sciences, the University of the West Indies. My primary expertise is pharmacology, especially looking at the pharmacology of plants and other natural substances and their impacts upon biological systems.
So what exactly is, you said it's the impact on biological system. Can you break down in layman's term what pharmacology is? You started, but can you?
That's right. So pharmacology is a study of the effects of substances on biological systems,
Okay.
In its shortest definition. But it is a very important and detailed basic biomedical science. And it is important because it interfaces the use of substances within
Hey,
not
Mike, how
only
you doing?
the
Good
clinical
night. What's up, man?
arena, which is
All right.
primarily in the healthcare facilities, but also in other arenas which look at the use of substances for household, personal care, among other uses within the current space.
How did you end up interested not just in pharmacology, but in pharmacology of the plants in the Caribbean and specifically in Barbados? You wrote a whole book on plants, of plants,
That's right.
specifically medicinal plants of Barbados.
That's right.
How'd that come about?
So again, you may have heard the story many times. As a young boy growing up, my mother,
Yes, Fifi, how
my
you
grandmother
doing? Thank you for
primarily
being here. What's
used
going on?
plants for medicinal purposes. One of the things I was accustomed to was the use of bush tea, specifically Cersei bush tea, right? And this would be given for various.
All right, guys, this is a video playing. Basically, this is a doctor from Barbados that's gonna really break down a lot about herbs for us. And this is interesting, is a lot of information. So you will have to get your cup of tea or or whatever you're drinking and just enjoy, because it's tons of information here for we to actually learn. And who knows, you might learn something and able to do something from what we learned tonight. So, let's go.
Various purposes. It was given to purge you, which is essentially to detoxify the body. It was also given if there were any viral or any other infective agents would have been considered to be affecting health. So I grew up within the space where herbal medicines were used by my mother, my grandparents, and of course, one of the things I observed was that there were not a lot of opportunities for the actual identification and development of medicines within the region. And I felt that we were in a very important and very rich area where biodiversity could be explored for the use of development of medicines. And I wanted to understand the process of the use of these plants, which were used in a very folklore setting as a bush tea, right, into a conventional medicine.
That is an interesting dichotomy that you just outlined. Folklore and bush tea, quote, versus quote unquote medicine. But you've also said that your natural, how you grew up, that it was medicine. So how did you end up as a scientist? And then when did that click, that connection happen? You said you realized that there wasn't a lot,
right,
that there was a lot of opportunity that we weren't necessarily taking. Just give us a bit of the path to going, that recognition, my training, this thing.
Right. So again, being exposed very early to the medicinal value of plants, of course, one of the things that I would have observed for myself was that these plants worked. So I believed in the medicine, right? And without highlighting too much of the ancestral influences which would have made such plants to be effective medicines, what I can say is that there would have been countless observations of these plants as medicines, even though it was not conventionally recorded within the scientific setting. But that propelled me to try to make a difference within our space and to build upon works of others, such as my predecessors in pharmacology, persons such as Professor Manley West, Professor Feng in chemistry, among other persons who were based at the University of the West Indies at the Mona campus to investigate plant-based medicines and identify the bioactive constituents of these plants, which provide these beneficial medicinal effects, but also understand the compounds to note that if there are other adverse effects associated with these plants, that these should also be documented.
Oh, so how long have you been the pharmacological guru of Bush?
Well, I don't necessarily consider myself a pharmacological guru, and yes, I am quite. I do have quite an extensive amount of time within the discipline, but I have been at the pharmacological exploration of plants for just about 20 years. Right. My study then focused on identifying anti-neoplastic or anti-cancer properties of medicinal plants that were popularly used in Jamaica, right, for anti-infective, anti-cancer effects. And what I essentially did was to try to validate those effects of those plants, which were pretty much known within the folklore space only.
So what are you doing here now? You're senior lecturer, which means you supervise students, that you're doing research projects. What are some of the things that you're looking at? In Jamaica, you said directly you were looking at anti-carcinogenic
compounds. That's
compounds.
Right.
So what are we doing in 2020?
So in 2020 Barbados, what has happened is since 2008, with me moving to Barbados, I have not only written a book on medicinal plants of Barbados, but I have dwelled into the development of this aspect of pharmacology, which focuses on natural products. And essentially, that itself is a niche area called pharmacognosy, which is the study of the actual medicinal value of natural products or sources of medicine, right? And I've developed a program. It's a graduate program, which is an MPhil PhD in pharmacology. And right now we accept students who have first degrees in chemistry, biology, pharmacy, biochemistry, essentially most of the biomedical undergraduate disciplines to come into this program and learn the basic pharmacology, which is required for them to understand how substances can be effective drugs and medicines, and then essentially allow them to explore various areas within the realm of pharmacognosy, which is within the scope of my research work.
That's a brilliant segue into the next prompt, because the cognosy part is about knowledge, and as you acknowledged earlier, there's a history behind this. So can you give us like a express train tour through pharmacognosy, through the world and through time in general?
Herbal medicine is the mother of all medicines. If you look in various sources of literature, it speaks about the history of medicine, especially medicinal products. They will tell you that herbal products would have been identified as being used as far back as 3000 BC. Right? And I'm quite sure other persons would have heard me spoke about cannabis, locally, regionally, would also note that cannabis itself was identified to have been used as far back as 3000 BC as well. So it could be that one of the first medicinal plant which was used would have been cannabis sativa, subspecies of cannabis thereafter. But let's now jump forward through our time series to AD, after the death of Christ. And some of the things that we have observed in the development of this discipline of pharmacology and also of medicine would be the development of basic medicinal practices within a conventional setting. And this is where herbal medicine moved away from just being indigenous medicine practiced by groups and pockets of people who were practicing the medicine because of observations that were made by themselves, into the so-called scientific space. So in the 15th century, what you had the formation of apothecaries, which are quite Eurocentric in terms of their development and establishment. But it should be noted that the knowledge base which formed the backbone of these apothecaries, which we would call in this day and age pharmacies or pharmaceutical type entities, would have been captured from various pockets of knowledge from around the world, all right? And we're talking about African uses of herbs. We're talking about Asian uses of herbs. We're talking about use of herbs by indigenous people from the Americas, both Central, South American, and even the Red Indians who were in North America, etc. In the 19th century, what we had was the establishment of the natural sciences, chemistry, biology, and then thereafter, we had the biomedical sciences, which then contributed to the development of pharmacology. So pharmacology was actually developed late in the process, once we understood the chemistry, the biology, and how the chemistry and the biology contributed to the biomedical sciences.
You have access to literature that was looking at, particularly in Barbados, the prevalence of the use of plant-based medicine in conjunction with what is considered conventional Western medicine. So the pill, as well as the poultice, the bitters, along with the drink. So what can you tell us a bit more about that? Just a brief overview of like what that study was, what the statistics show, and then how that prompts kind of how you direct your research.
Yeah. Currently in Barbados, through studies that I have done, and also corroborated by other literature sources, the use of herbal practices in Barbados is just about at a prevalence rate of about 33%, right, of the population, right? So it could be said that approximately 33% of the Barbadian population actually access herbs for medicinal, herbs and other plant forms for medicinal purposes. Interestingly, within that group or that demographic of the population, we find that that women, right, the females tend to be, more advocates and users of herbs. And that ties in quite well with the role of women in society as mothers, as the caregivers of families. We also found that the knowledge permeated among the older group of that demography, of that specific demography, which shows that the retention of knowledge is actually in that older bracket. And if we don't find useful ways, such as a documentary series or written texts or books, we are at the risk of losing that information because young people might not be as exposed to that information as the older folks may have been. Other interesting and useful facts about the studies that we have done in Barbados is that the plants which have been identified for medicinal uses, 66% of these plants have pharmacologically active components which can bring about beneficial effects. But again, one must always place caution on that statement because it has, while they are pharmacological active agents produce beneficial effects, some of these compounds may have toxicities or could bring about adverse effects as well. 51% of the plants which are identified in folklore for their medicinal uses have phytochemicals that have been fully validated for the medicinal effect that is actually reported among folklore practitioners. And that is important because it speaks to the fact that the use of herbs, even in its colloquial or very natural and traditional space, is actually scientifically validated.
So we're in our science superhero personas now, lab coats and all.
That's right.
And we're going to get very practical talking about
Yes.
the preclinical drug pipeline.
Correct.
What is a drug pipeline?
Right. So the drug pipeline is essentially the pathway for the development of compounds from their very natural or synthetic sources all the way into clinical medicines.
So it involves the science of it, but before that, you have to kind of even know about where to go and look for these medicines, right?
Correct.
So can you tell us a bit more about, we're going to get into this specific plant, but perhaps people in Barbados who have done it before or where you all look for original sources and historical sources to kind of, let's investigate that plant.
Yeah. So the again, going back to the drug development pipeline, it's primarily in three components. And because we are a resource restricted institution, and I guess nation, we are not able to complete the full drug development pipeline within our current setting. But the three important components of the drug development pipeline and the parts of those that we're able to commit to within this setting would be drug discovery, right? Preclinical development, and the last component of drug development that we can't do or we don't extensively do within our setting because of resource restrictions would be clinical development, right? So as it pertains to the actual identifying of these plants for use, that would fall under drug discovery, right? Which would have essentially provided information, which would have propelled, and inspired an interest for the investigation. And there are many sources where we gather information. As an academic, it's part of my role here to do research, information gathering. And information gathering can be very formal or it could be informal.
Let's talk about the informal one.
I know that was where you wanted to, right? So informally, through myself and other researchers, we would have had opportunities to speak to herbalists, right? We also know that there is a retention of knowledge about herbal practices by the older population of Barbados. So apart from capturing that information in surveys, we'd have had focus groups, we'd have had opportunities to hear from persons about these uses. I have, in being able to compose medicinal plants of Barbados and to also publish in the area, it is one of the expected interactions that you must have. You must reach out to persons who have practiced this science And at this point, you hear me mention herbalism as a science, because persons tend to think that it's not scientific, but it's very much scientific in its approach. Because if we look at the most basic definition of science, which is actually recording repeated observations and drawing inference, persons who would have captured this information prior and have used herbal medicine within folklore would have made many observations of not only their grandparents and foreparents and ancestors using these herbs, and have seen that there would have been some beneficial use and that they were also safe. Right. And that itself is science, because they have made inferences based on countless observations. And that is no different than what we do in the formal scientific setting, where we actually document the information, right? And we use statistical analysis, right? To outline if something is statistically significant or not, or clinically significant or not. But it's the same process that our foreparents and persons who practice folk medicine would have used to actually have reached this point.
So the lore, these folk stories, or because we tend to capture a lot of things in a non-straightforward way. We're talking about medicine, so I will not mention that cocoa tea. But when you listen to songs, etc., there's clues in there, and you're saying that is not outside of the pale of something that would prompt you to go and look at that particular compound or that plant.
Exactly. There is always a scientific basis in folklore, right? No matter how basic it is. Because what we have to understand, even when I spoke about the time series of, herbal medicine, right? Or just the use of medicinal plants as medicines, we started from very primitive knowledge and sources where herbal medicine was at one point considered black magic or just magic, right? We had persons who were considered witches and sorcerers because they practiced herbal medicines, and they were staked because they were considered to be, outside of the conventional society, which was more Christian based on the Roman Catholic Church, etc. But that science itself was not well defined. And what we have been able to do in capture that information from all these various sources, informal and formal sources, is to find a way to validate the science. And let me explain validation. So what we're doing in the lab now is validation. But validation can also occur outside of the lab, where you find quite a number of plants. They're not only used in one geographic location. I can tell you that none of the medicinal plants in Barbados are considered indigenous or endemic to Barbados, but they're used in other Caribbean territories. But when you look at the medicinal use, the benefits that are reported to be aligned with the use of these plants, they're quite consistent. So that itself is another form of validation. So we look at the medicinal use, the folklore uses of these plants in different geographical locations, and we don't only limit that to the Caribbean, but we also look at West African practices. We also look at East Indian practices, right? And we also look at the practices by the indigenous people who were within the Caribbean before our colonizers came. Right?
You've got this book here, which is going into another edition. It has an extensive bibliography.
Yes.
Which itself speaks to the tradition here of people with botanical gardens and of herbalists and scientists doing this type of work. Who are some of the people that have inspired you or that you use for reference?
Well, I can tell you, there are two key persons that I consider to be very instrumental in assisting with the information that has been collected in this book, and would have assisted in my ability to do this work in Barbados, focusing on medicinal plants of Barbados. One, of course, is Sean Carrington, professor of botany, who is the local expert on plants, right, of all types in Barbados. I was also shared information by other horticulturists and other researchers, before Sean and my time, such as Iris Bailey, who wrote extensively, on the use of plants, right, and practices, the cooling teas or bush teas of Barbados. But I've also worked with other persons in the field who are, more within my time of my research, such as Sonia Peter. We did share some time working with a student, Sheridan Carrington, who was a co-author for some of my published articles, etc. So it's a community, right? And it's because you need a botanist, persons like Sean. You need a Sonia Peters and other natural product chemists, to be a part of the process because they can go through the chemical extraction process quite well. And you need a pharmacologist, right, who can then, take these plants and the compounds from these plants and put them within the medical context to say, okay, let us explore the medicines of these plants by going through this step-by-step process, hence the drug development pipeline.
All right, so let's
go through the pipeline.
Jump into the pipeline.
So
what is this beautiful bouquet?
Well, well, this was harvested just for you, right? But
thank you.
But as you can see, it's a quite popular garden plant. I'm not sure a lot of persons understand the history behind the Madagascar periwinkle. This is not unfamiliar in our territory. This is not unfamiliar in other territories in the Caribbean. We see it and know it as a garden plant, essentially, right? But there's a lot of value in this plant, right? And let me explain what I mean.
Oh, sorry. Welcome, everyone. Sorry, I never realized I was mute. Thank you guys for being here. Appreciate you guys, man. All right, so now he going and show us how he get the medicine from the herbs. Man, this is interesting, and it didn't look too complicated. Check it out, guys. Check it out.
I mean by that. In the 1950s, this plant was being explored for anti-diabetic properties within the Caribbean setting. The research within that setting showed that the compounds extracted from this plant had other uses. They realized that the compounds from these plants were cytotoxic, which essentially are toxic effects occurring in cells, right? Which would lead to lethal effects in cells, the killing of cells, cytotoxicity. Cytotoxicity is one of the main mechanisms that are actually explored in the development of anti-cancer drugs. And from this plant, there are two well-established, westernized, or conventional anti-cancer drugs that are actually used currently to treat cancer.
Okay.
Right? Vincristine and vinblastine. Right? So a lot of people don't know that they actually have a gold mine sitting inside the garden, and hopefully this is not going to inspire, inspire people to go on. Well, because I've heard questions. Persons have asked me questions about, doc, if I want to plant, I get diagnosed, and they come and they're very confidential with information. And of course, you have to be empathic, but at the same time, you have to be real with them. They said, well, I was recently diagnosed. Are there any plants, herbs that could be used? And I know that there are herbs that can be used that have compounds which have shown to have these important properties. But because of the lack of standardization, right, of the compounds and the dosage, right, and again, because we're not fully, understanding all the various adverse effects and toxicities, which might not only relate to the anti-cancer compounds, but other compounds in the plant, we like to encourage persons not to dwell too much on using these plants, especially for conditions such as cancer, which, as far as I'm concerned, if the treatment is not evidence-based and standardized, could cause problems in terms of outcomes, right? Morbidity and also mortality.
So because of that, and I know that what we're going to be looking at here is how you are trying to standardize the phytochemicals and the bioactive ingredients in them so that people can take 2 teaspoons or a drop or etc. So how do we go from this then to some of those things?
Right. So, so again, to make sure that we're on path of our drug development pipeline, so we're still in drug discovery. We got information that this plant, has some very useful properties. It's not only anti-cancer, but it's also anti-diabetic, and it's also hypotensive. So it can be used to manage hypertension, to low blood pressure as well, right? So you start with your harvested plant, which is fresh, as captured here. And the next step really is to make sure that you have the correct identification of the plant. And this is where you now need to work with a botanist, someone such as Sean Carrington, with his extensive background in being able to identify and participate in applying nomenclature to new species of plants. So that work is done within an herbarium. And for the purposes of our research work at K-Ville, in the Faculty of Medical Sciences at K-Ville, Sean is our person or the resource that we use as the herbarium. to identify the plant. Now, the identification of the plant is
Hey, boy, Blue. I think the name of the plant was on the screen. Yeah, I think that I didn't get the name. I didn't really see the name, but I think the name of the plant was on the screen. So maybe if you go back a little, just a little, you might see it. The name now come off the screen. I think that's the name for the plant. So just go back a little, and I think you're gonna see it on the screen.
is important because it's important that we know the source of the plant as we go through the drug development pipeline, right? So after Sean identifies a plant, the next step, of course, would be the preservation and the vouchering of the sample. So what you're seeing here is actually a preserved sample, same plant, but preserved, right? And it's vouchered. And vouchering is important because every single identified species has to be vouchered so that as we continue the drug development pipeline, we can always trace the source.
So it's your paper trail.
That's your paper trail.
Okay.
That's right. So the preservation is important because as a part of the thesis and in the written document or publications to come, you have to outline this process quite clearly.
Okay.
Right? Okay.
So this is it dried. This is how you preserve it.
So this is it dried, but not only is the drying important for the preservation, but as we now move from harvested plant in its, I guess one could say it's hydrated or wet form, towards the extracting of compounds, a very common practice based on the nature of compounds that we tend to look for is the drying of the plant. Now, the drying of the plant, apart from drying it for preserving the plant, can be done in the external air. Only problem with drying it in external air, of course, is the fact that you run the risk of getting the plant infected by all sorts of different infectious agents that may be in the environment. But it could also be dried in a solar dryer.
Okay.
Right, which accelerates the drying process. Drying is important because what you're now doing is you're now starting the process of standardizing the plant material to carry through the extraction process, right? And eventually all the way up to
making the chemical.
To making the chemical, or well, extracting the chemical and making the drug, right? So you dry the plant. Once a plant is dried, and drying basically removes moisture, water content from the plant, right? Sometimes drying, dependent on the external temperature that the plant is placed in for drying, may remove volatiles. Now, if volatile compounds are not within the scope of your interest, then that's not a problem. But if you don't know what compounds you're looking for and you remove the volatiles, chances are you may lose some of your biological activity, right? But we tend to go with the dried format, and we dry it in the open air. We just make sure that the area is very secure, right?
So we've got our vouchered, preserved sample. We've identified it. We've talked to the people. We listened to the songs. We found the plant. We identified it very specifically. We've dried it. What do we do now?
So we spoke about drying for two primary purposes. One of which was for preservation, I guess part requirement for the thesis or the research project. And two, we spoke about drying because of the important need for standardization of process, right? So the next step, of course, is to move into creating that material now in a material which would essentially facilitate the extraction, right, of the specific compounds that are required to carry out the necessary drug development pipeline. This here, what you have is a miniature mill, right, which can be used on your desktop. It is on our desktop. And what it is, essentially, it's a very sophisticated, minute piece of equipment which allows us to take the dry plant material. And again, dependent on the part of the plant, which has been reported in folklore to be useful for the medicine effect, because in some of the literature that may exist, inclusive of my book, it will tell you that it's sometimes the leaves, it's sometimes the stems, it's sometimes the roots of the plants, right? Then, you know, those dried components of the plants are placed into the mill. And if I could just turn it on just to give you a quick demonstration of what the mill sounds like. It's nothing too extravagant, Right, there you go.
So we put the material in here.
Right, minus your finger.
Cover
Right.
it down.
Yes, right. And as you can see, the rotating arm inside this section here, which is guarded by the glass plate, essentially will then interface with the plant material. We would use a pestle, a wooden pestle, to essentially force the material all the way through the funnel until it gets into this chamber, right? And then the material is milled. And of course, we will have a jar like this. As you can see, this one is labeled Cersei. But if we're milling Cersei material, dried Cersei material, we will have the jar underneath the receptacle here. And as the plant is milled, it will be collected in this jar. So this is what the final product looks like for your dried milled material. So you move from right, and you can get the essence, right? The compounds are ready to be extracted. You can smell your Cersei,
Yes.
right? And this is not periwinkle. We started with periwinkle, and there are other samples such as this periwinkle sample, which has been dried for some time, so it has been oxidized to some extent. So that is your mill. And as I stated, what you're trying to do, you're trying to take the dry plant material and convert it to a material which will now allow and facilitate the extraction process. I couldn't move to the next section without identifying and enlightening this very useful piece of apparatus. And this was what the equipment was like in the early centuries, in the advent of medicines from plant material. So this is your mortar and pestle.
Right.
So your pestle and your mortar, and we do got some dry leaves of the periwinkle,
Okay.
right? Already in here, and again, you can use your mortar and pestle to break down the plant material, right? So that when you add your solvent, and solvent in this case could be the medium that you want your compounds to be extracted in. So it could be a chemical solvent, or it could just be water.
Okay.
Right. Note also that you can extract wet plant material or undried plant material. Again, persons who develop poultices develop their poultice in a mortar and pestle, and poultices are used to be are essentially used for topical applications more so than the teas and other extracted forms and capsules, which are used for enteral purposes, which means oral route, etc.
So we've milled it. We've got it in these really. Can I touch this?
Of course, yeah, you can touch
These
them.
really cool. Your periwinkle, your seed under leaf.
Right.
This looks like my tea jar.
That's right.
Cersei.
Cersei.
And then we get to do a lot of very interesting stuff. And we're moving over to
the Soxhlet apparatus. So what the Soxhlet apparatus is, and it looks very sophisticated, but what essentially is, it's really just a very unique and highly resourceful mechanism of capturing the extract of the plant material in a very concentrated way.
okay.
Right? So there are two scientific principles that are taking place here: distillation and percolation. Right? Some persons use the terms interchangeably, but within the mind of a pharmacologist, they're separate, because we have percolation that is occurring within the Soxhlet, the main element of the Soxhlet here, which is a central location. This is your condenser, which facilitates the distillation process as the solvent is heated in the round bottom flask. So the whole unit is on a heating mantle,
Right.
right? And make sure I get the
So
H in.
the
as
hot
you make-
plate.
Yeah. So the hot plate, right? And applying heat to the solvent, right? When the temperature reaches the boiling temperature of the actual solvent, or the boiling point, right, of the solvent, then it evaporates, right? Upon evaporation, of course, it will go through the Soxhlet, the large arm,
Okay.
right, where we will then essentially meet upon the condenser.
Right.
The condenser is called a condenser because it actually will lead to the condensation of that solvent, which has evaporated, right, or the vapors of the solvent. So upon the condensation of the solvent, it will drip into our plant material, which is in the central location of our plant material. So the plant material within our Soxhlet would be the milled periwinkle, or the milled
So
cerci.
this is no milled periwinkle in here.
There you go.
Yeah.
So percolation is actually occurring now. Percolation is just a process of passing solvent over the plant material, over the milled plant material. And some persons might be asking, why did you mill the plant? Was it just so that you can fit it into the sucklet? But no, by milling the plant, you increase the surface area of extraction.
Right.
So you can get a more efficient extraction process. And what happens is that this process is a cyclic process. And what you end up with in your round bottom flask is a concentrated volume of extract of a specific solvent.
So this green thing will eventually end up
There
here.
you go. As it continues, as the condensation continues, and we have sufficient extracted solvent of the plant material gathering within the central unit, it will go through the smaller arm, right back into the round bottom
But
flask.
it won't do that until all of this has processed through.
There you go. Until the solvent would have arrived at its boiling point or boiling temperature to essentially facilitate evaporation.
So then when we have the concentrated matter, because you distill the water, then it's percolated back to it. I'm thinking of coffee, I'm sorry.
Yeah.
And it comes back to it.
There you go.
And
You're
now
percolating in the kitchen. It's the same process.
So you have your liqueur,
Yes.
the bush.
Mhm.
Then what do you do with that next in this pipe, in this pipeline?
So having gotten your concentrated extract, and some persons might be saying, are all plant extracts green? No, they come in a variety of different colors. And this might sound a tad bit scientific. So remember, the color of your extract is just the wavelength or the specific wavelength at which the plant compounds absorb light. So the green you're seeing, it's because it's actually capturing or absorbing wavelength of light within that spectrum of the color sequence, which would actually depict green.
And not just because it was green.
And not because it was a green plant, right? So we take our extracted and concentrated plant extract, and it's important that we really concentrate the extract, right? So that we can identify these compounds, and it's also a part of the very important standardization process. And I know persons might be listening and say, well, I always heard the medical doctor say that the use of herbs is not standardized. So are you telling me that I have to do this in my home setting? No. But it's important for us to do it in the scientific research setting, because as we move on to test these compounds, we have to be able to show traceability and also dose response relationships. We don't expect persons out in society to do that on a day-to-day basis. This is something that is quite preserved for the research lab. So we move to our rotovap, which essentially is using some of the technology that I would have described on your sucklet apparatus. And essentially the rotovap, it's a water bath with water, right? With distilled water, so that we don't get a lot. It's right now it's cool, but if we were to notch the temperature up, we should be able to increase the temperature. As you can see, the temperature dial is now saying 70.
Okay.
So eventually, as the temperature increases, you will see the actual temperature of the water move towards what it's being set at. So again, what you're doing here, you're going to set the temperature to the boiling point of the solvent which was used. So we're using hexane, right? Which is a nonpolar solvent, and the boiling point for hexane is not excessive. So if we were to set our rotovap or the bath or the temperature of the bath just about the same temperature as the boiling point of hexane, we should be able to remove the excess hexane from the actual
so this
extract.
is a further purification process,
right.
So that
or isolation, or more along the line isolation. What we're trying to do, we're trying to remove
even
the
more
solvent.
concentrated.
That's right. We're concentrating
of just what's in the plant
and isolating the chemicals from the plant and removing as much as possible the solvent that was used to extract the plant material. So that is essentially what takes place here. And again, you're seeing some similar components of this equipment to our sucklet apparatus for the initial extraction. Again, another condenser, because as we arrive at the boiling point of the actual solvent and it goes into our condenser, it will actually condense, and then we capture the solvent which has been pulled off through vaporization into this round bottom flask. Right? So what we end up here is a very concentrated plant extract, which can now move on into the drug testing and drug pipeline process, as described earlier.
Okay. And now that you've isolated as much of the plant extract from the solvent as possible, that's when you then start looking at what's in there that would be bioactive.
There you go.
In the preclinical trials.
There you go. There you go. Well, before we get to preclinical, which speaks about the use of animals, right? There are some other important steps in drug discovery, because we're still in drug discovery. We're not into preclinical until we have, shifted our focus into animal-based testing. So, so within drug discovery, which is the broad category given to that very first component of the drug development pipeline, you have, lead finding, lead optimization, right? So we're still into the lead finding aspect of the process, because what we have now done is that we have now identified and extracted some compounds, right, from this plant extract. We now need to go into the chemical identification of these compounds to know exactly what we're working with. So that is now.
So when you pour it from there, what next?
So we take the concentrated plant extract from the rotovap, and then we carry it over to our bench shop. And it's important for us to get an idea of the compounds that are in that extract. And we do this preliminary by a process called thin layer chromatography. In the most basic sense, what that involves, it involves a stationary phase, which is an absorbent type material, in this case silica, and then a mobile phase. Now, through the extraction process, we must know that there are a variety of compounds in the plants, and some of these compounds are what we describe as polar compounds, and some of these compounds are non-polar compounds. Through the extraction process, and to be complete with the process which was described earlier, when we're extracting plant materials, we like to go from very polar solvents to very non-polar solvents through the extraction. So we have different solvent extracts. So the next step is trying to identify if we can separate some of these compounds by using a mixture of solvents, which we will now consider our mobile phase. We already described that the stationary phase is already on the TLC paper, and that is silica, right? So we have our TLC jar, and what essentially would happen next is that some solvent is placed in the jar. We would take our TLC paper, so we'll move from a large sheet like this, and we'll cut small little cute rectangles. And what we will do, we will take our extracted material, and we will make spots, right, on a line on the TLC paper, which would make the first contact with the solvent.
Okay.
What should happen is that the solvent, upon touching the TLC paper, and the solvent, and the silica itself being absorbent, it should allow for the solvent to move along the stationary phase.
So in my mind, watercolor painting, I put the pigment on it.
That's right.
And then when the liquid comes up,
it
makes a streak. And you're saying that the different colors in the streak are actually separating the different compounds.
That's right. Sometimes you won't be able to identify different colors on the TLC paper, but what you'll see different bands. Right. And each band would essentially represent a group of compounds. In some instances, dependent on the number of compounds that was extracted, may represent one compound.
See, integrated science did teach me something.
There you go. So this is what takes place in this jar. So you have solvent here, and some persons are saying, but if you do an hexane extract, are you going to put back hexane here and think that it's going to cause any change in the compounds ascending the TLC paper? No. So we wouldn't use hexane by itself, but we would use hexane mixed with, say, a more polar solvent than hexane, because hexane is the most non-polar of the solvent polarity series, right? So it could be a dichloromethane, or it could be an acetone mixed with some hexane. And what you intend to do is to separate these compounds as that mobile phase start crawling across the stationary phase, which is silicon, the TLC paper. Why is that important? It is important because you have now identified that there are different chemical groups, right? And to make your scientific testing a lot more rigorous and a lot more specific, it's important to now capture those chemical groups separately, right? So in order to do that, we then move to an actual color.
Okay.
Right? So the column is essentially a larger unit of what I just described. It has a stationary phase with silica, right? And then it will be introduced to a mobile phase. Unlike the TLC paper that was the mobile phase was at the bottom of the TLC jar, we're going to add the mobile phase on top of the extract, which will be placed on the silica. So if you look at this and you look at the various colorations, and just to give you an idea, and this is not perfect, but this is just for demonstration purposes. This is your silica. So you'll pack your column first by stopping the bottom of the column with some cotton or some wool. Then you add your silica, right? The amount of silica added will, of course, facilitate greater separation of compounds as they move through the column, right? And then you apply your extract. So you're saying, but this extract here looks solid and it was liquid there. Well, having extracted the compounds and concentrated it into a small volume of a solvent, before you can apply it to your column, you have to place those extracted compounds in a solid form. And how we go about doing that is that we take the very viscous and concentrated extract of compounds.
Thick and strong contrast.
And we mix that with sand. Sand, right? Activated, clean, purified sand to make a specific solid mass, which is then applied to the top of the column.
So a little pill, like.
Basically, right? But and it's so that's what it's going to look like in the column, because it's going to sit right on top of a silica, and then and that has a very unique and important process. One, it prevents the extract from being washed within the silica as this mobile phase is poured, right? And it keeps the extraction and the separation. Sorry, it keeps the separation and no longer the extraction of compounds from that solid mass that has been applied, very unique and uniform throughout the separation process, right? So when you add the mobile phase, what you're going to see as time progresses are different chemical bands. Now, for each band, and you will visibly see these bands going through your silica, right? Again, because we saw that that was green, we will see a very concentrated green band. We might see a yellow band. We might see, you know, a orange band, right? You know, so those are the consistent colors that we tend to find with the chemical groups that absorb light from plants. So you have your column, and what you're seeing here would then reflect different bands that have been captured. So each of these conical flasks would have a separate band, and what each band represents, again, could be one compound, or it could be a group of compounds, all belonging to, well, it could be one compound, or it could be more than one compound belonging to the same group or chemical group of plant compounds. And earlier I described that you had various chemical groups. You had the flavonoids, you had the terpenoids, your anthraquinones, tannins, etc. So it is these fractions that we actually test, right, within the biological systems. And then...
So this is not what you're meant to go test.
There you go. So this is essentially what we test. But before we test, because we don't want to test these extracts and solvents, we have to, again, concentrate them, and we carry them back to the rotovac.
Right.
We pull off the solvent.
So science is a lot of rigorously actual work.
There you go. But the chemistry is very important. But what the chemistry does, the chemistry highlights the need to be able to identify different compounds within the plant extracts, different groups. And what else it does? It allows for a certain level of specificity of the testing that follows the extraction process. So in making sure that we connect the dots, so in the drug discovery, we'd have gone from identification of plant, the drying, the extract, the milling, the extracting of the plant compounds. We now have the isolated compounds, right? And it's these compounds that we then test on our specific targets.
All right.
So our drug targets within this lab, as I pointed out earlier, would be based on the disease forms that these targets impact. So we do work on diabetes. So we look specifically on drug targets such as the DPP4 inhibitors, which are your dipeptide peptidase 4 inhibitors, which, as I said, are important in leading to increase or enhance insulin secretion in diabetic patients. And for hypertension, we are very much keen on looking at important enzymes like nitric oxide synthase, which produces nitric oxide, which plays an important role in the relaxation of our blood vessels. Right? So those are enzyme targets, but they are very important drug targets for us to focus on.
So drug targets from plant,
To
So
isolated compounds.
there you go. Compounds from our plants to specific drug targets within the biological system, right? Which will then allow us to be able not to say that periwinkle or Circe has that effect, but we can then speak specifically to the type of compounds within Circe and periwinkle which has that effect. And remember, we're still in the pipeline. So it is important for us to understand that at this point, we are just moving from lead finding into lead optimization, because we will have different compounds in different fractions, and we will see that some compounds in some fractions have greater activity than some, which is now moving into lead optimization. And we have to look at the specific type of compounds and the medicinal effects that they have, because you may have compounds which may have beneficial effects, but they may also have adverse and side effects. They may be toxic compounds.
You have to give you your own series.
So you don't want those compounds, but what you're looking for a compound in a different fraction that will have that medicinal effect without the side effects. So lead optimization. And then from that point onwards, you go into your animals, right? And then in our animals, we essentially test that biological effect to see if the medicinal effect, the beneficial effect that we saw on the drug target in the in vitro space, which is looking at the enzymatic assay or looking at ex vivo assay within a cell line, carries over into the living organism, which is the animal.
Okay.
Good.
Thank you.
Yep.
All right. What you guys think, man? What you guys think? What you guys think, man, about this? This was a really, to me, it's very interesting. And what I like how he break down the whole process from the plant. That's really exciting. That's some real stuff there. So I got to shout out to NCF Barbados for this information, man. This is great information you guys putting out. I got to say shout out to you guys putting in this kind of work. Oh man, this is amazing for real. For real. I think it's a very interesting life. I really do. I really do. It's interesting to see the progress. Well, the process. Yes, it really is. It really is. At least, because we don't see these things. We don't see, we only hear about, yes, 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 yes. We only hear about the hubs, we only hear about this hub do this for you, but we actually never see, no, never understand the breaking down and the process of how it happened and how it works. And believe it or not, the one he have this pretty simple. I'm sure by now they have more advanced type, but whatever as simple as it is, it seems like it doing a great job. And what I noticed in times gone by, like in the island, I remember when people used to, and that show is in Barbados. Barbados is an island in the Caribbean island. That's in Barbados. Barbados or whatever. But I remember Barbados always have like the more advanced for Caribbean island at them time. They were like more advanced in what, because I remember when people used to leave from different islands going to Barbados to see doctors and stuff. So it seems like they put a lot of effort into medicine, and this is what's important. This is what's important. And it's crazy because we have so much, sometimes we have these plants actually growing in our garden and in our backyard, by the side road, seeing them and have no idea that these plants is the plants that grows to actually help us. I don't know I found a plant today, and let me show you guys this plant. I have this plant, and it's a grass. But I have this plant growing it's grass. It's like it's a cover. You could have it in your yard and it will keep down. It grows up real nice and it push out a nice pretty flowers. But also it push out a berry as well. This plant, you know where you could plant this, like you have a bank, like edges, you could put that by in the edges, like shape, put it real nice. And when it start flowers, it so beautiful. But I never know that this plant has so much benefit. And it really is. Let me see, that's true. My brother was a baby, he has a stomach. So exactly, exactly. Barbados, like they have, they had the thing going, man. Cause like Fifi now say, and I know that used to happen in times gone by, where you used to take people used to go to Barbados to do surgery and them kind of stuff. But there's so many plants. Let me show you this plant. Maybe this plant maybe you might have this plant growing in your backyard and you have no idea. So let me show you this plant. I did a video with it. So let me show you the video. The name of the plant is dwarf lily. Give me 1 second. Welcome back. Searching for herbs, searching for different type of herbs. We run into this plant right here, and this plant, the name of this plant is dwarf lily turf. Dwarf lily turf. As far as we understand, this berry is very good and have lots of benefit for children, for the lungs, for skin. But guys, this is just for informational purpose. Please do your research. Please speak to a doctor before proceeding. Look at the plant itself. Look at the leaf. Look at how the leaf looking. Who knows? Maybe you might have this in your backyard. But please do some research on this plant. It's dwarf lily turf. That's the name of it. This is how they look when they green. This is how they look when they green. It produces a berry. Let me show you a few more. This is a next one right here. Nice bunch of lily of grapes. You see, this is it right here. Look it here again. I'm sure if you look pretty good, you're gonna find this in your backyard. I'm sure a lot of people have this in their backyard and have no clue what it is. So like I now mentioned, guys, the name of it is dwarf lily turf, and this is something they use for medicine. Do some research on this plant, and please let us know in the comment section if you see in this plant, or if you have it, or if you have any information about this plant. Thank you guys. I hope you guys enjoy. Okay, is any one of you know that plant? Yeah, you see what I'm saying, Fifi? Yes, and I have this growing like crazy for years, up and years. I never know that this plant. You see, this is why I'm saying I got to start doing my searching. I got to start walking with my book. I got to start walking with my phone, take pictures of these plants. The plant is a powerhouse. As simple as you see it there, it's actually a powerhouse, that plant. But guys, as usual, disclaimer, please speak to your doctor before using that plant. Because I'm not a doctor, I'm not giving medical advice. All it for is for informational purpose. All right? Let me see. Okay. If you say that's so true. When when my brother was a baby, he had a stomach surgery in Barbados, even up to 30 people. People still travel there up to 30. So it seems like Barbados is doing pretty well with their medical. Like they invest into their medical, they invest into their hubs. I'm hoping that more islands could do that because they have so much hubs down there that maybe it's the animal eating them, getting the benefit of them. You know what I'm saying? When the rate of diabetes is so high and you have food, you have herbs that could actually help you right underground. Come on, growing all year round. At least we got to, people in different country were dealing with the winter, it's seasonal, some of them. But you have them there all year round. You have the weather for that. Whoa, that is a really good plant, yes. It's a Chinese medicine, is good for the heart, lungs, spleen, and it exactly calm irritability in the body. I have so much. I'm telling you, right by you see me was there. That's the whole era full with them. Every year, and I'm cutting them. I'm load of so much benefit. This is for real. Yes, it also called mondo grass. Yes, it called mondo grass as well. And the thing is, this grass, it spreads. Like I said, you could put it by, like you are, say you're doing a flowers era or a garden era, and you want to use it for like do the barrication. You could use the grass and do the barrication around the bed, and that will just grow around the looking beautiful. Then it push out it beautiful flowers. Then it push out it beautiful berry. So what I'm gonna be doing now, I'm gonna be taking care. I actually know. No, it put me in a vibes now where I'm gonna actually be taking care of a portion of them, and picking all them berries. I mean, and as you see in the video there, you see it's a lot of berries. It's plenty berries. Yes, can't grow it fast enough when sick. When sick, grow it now. Got you. That's it, man. That's it. That's it. Can't grow it fast enough when sick. Grow it now. Makes sense. Makes sense. Most all go dry as teas. That is for real, man. You see, this is what I'm saying, man. This is what I'm saying. We come together and we could actually learn a lot. We could, you see, because this is what's going on here tonight. Simple, so within this space of time, I've learned a whole lot. All right. It helps with anxiety, good for liver, and a whole lot of things. It's really a super grass. It's pretty too. Yes, You want to see it. You want to see it when it flowers. Oh my goodness. You want to see you have a patch of it. And the beauty of it, man, listen to me. It push up a beautiful flowers. Pretty. You know what I mean? So if you have a line of it in a nice, all you just seeing that beautiful flowers, man. It's a beautiful plant, man. Let me see. I look into it to grow inside. Oh, just give me some. Just let me know how that go for you, Mike. Let me know how that goes for you. Let me know. But and it do for real. It do every fall. Every fall, it produce this. Well, now it's fall and you see it have the berry. In the summer, it had the flowers, the beautiful flowers, man. They're pretty, pretty. And I never know these things loaded with benefit. Power plant, for real. Thanks for that planter, another new hope. Happy, thank you, Fifi. Thank you. Yes, I this is what I try to do, man. I try to once I see it, I'm not holding it to myself. I'm share, you know what I mean? I'm sharing. And it's good to able to have have people that you could share stuff with and they could share with you, because I'm also learning about plant from you guys as well, which I appreciate that. And I'm a person who very into herbs. I'm very into herbs. I'm just sorry that I didn't start out earlier this year into my journey of looking for herbs. Because now it start getting dry down now. Everything start, you know what I mean? So I'm sorry I didn't take up that journey. Maybe at least in the summer, start looking for herbs, because now I'm looking now, and believe it or not, I'm running into some herbs. I'm really running into some herbs. I'm even seeing herbs that I see in the island, that I actually seen in the island. Yes. Yes, Fifi, I love learning new plants. All right, all right. That's that's good. That's good. We all do. For real, we all do. I have a spot in mind to grow some. Well, believe it or not, everyone should grow some. Everyone should grow some. For what we getting for understand, why not? Okay, why not? A lot of people use it for landscaping, not knowing the benefit. Exactly, Audrey. Exactly. They, because, like when Mike say, yeah, monkey grass. Yes. They have different name. I'm researching now. Definitely. Grow it between plants as ground covered. Definitely. That's what it do. It grows and it's kind of spread. It push up plenty. Then when you do look, you see the spreading out. Ah, listen to me. I have a big oak tree. It's one of the biggest oak tree I ever see. The biggest one and the tallest one I ever see. An oak tree. And they spread right around the oak tree. Right around. If you see how they spread out around the oak tree. And oh my goodness, when you see them thing push flowers, when you see they push out their flowers, if you see the beauty, you know what I'm saying? Then after the flowers, you see they push out this blue, this dark, rich color, blue, almost like purple, almost look like black. When you see they push that, man, is so now I have something else to look into. Mhm. It kills weed. Yes, it surely do. It surely do. Monkey guy. All right. Yes, yes, it's a very interesting plant. When I study the plant today and look up the plant and I realize, damn, the benefit of this thing. I did a short video. I did a video putting it out there because I'm sure there's people have this grass growing and have no idea of the benefit that these, and like what you know say, a lots of people grow it for landscaping, but they don't know the benefit of the plant. It's done 6 to 10. Beautiful plant, man. Let me see. Hold up. Okay. Oh wow. Oh wow. Okay. Hold on. I'm trying to get the it's almost look like let me see. Okay, give me 1 second. We might say so. Hold on. It's from 6 to 10. Okay. It's okay, all right. I I can't type tonight. Okay, all right. Yeah, let me see. Hold on 1 second, guys. Let me see. Okay, let me hold on. Let me see. Yes, this is basically this is it right here. When you see this thing start growing, you see that kind of flowers, man, beautiful, man. Have you and if you have them shake up really nice, which is what I'm gonna be start doing, yes, beautiful plant, man. Plant are like, let me see. You like my sweater? All right. Little chilly tonight. I was outside, so I had also pull on a sweater, boy. Don't know you go already. Can't allow the weather for pull you down. All right, thank you, Boxy. Yes, guys. Yes, yes, yes. So tonight was very informative because we learned a lot. Yes. Let me see. Boxing man, I take that stand 50. It's 32 there. My goodness. Wow. Man, you ain't even in the winter yet and you dealing with oh man, that's some serious weather for real. Man. Yes, it reminds me of Ted. Okay. Like you. That's some man. I don't know, man. That's some serious weather you talking about, there, man. I know. Honestly, I know. I walk in some weather, 30 degrees, 28 degrees. Listen, me. I walk in some weather already. It's only left for me to cry, man. I'm telling you, serious weather. So I kind of have an idea of what you're talking about. But my goodness, that's some serious cold weather you dealing with, there, man. Wow. Snow starts soon, like 4 hours. Ooh. Mm, I remember shoveling out your car. I remember that time when you wake up and the snow truck pass and the snow truck push and when you come up, the snow is all cover up your car, the side of your car. You got to use a shovel. You got to dig out your car. Your finger them freezing. I remember them times. So I have an idea what you're talking about, Mike. Damn. Mm. Some serious weather you talking about there right now. Yes, guys. So yes, man. Yes, it was very interesting. I have a few more plans that I have to upload. Few plans I got to upload that I also found. Pretty interesting plan. I'm gonna keep doing that until. But I'm just sorry I didn't start out earlier, for real, because I miss a lot of plan, for real. My husband walking zero, damn, weather sometimes. Whew, man, I think I did my time. Well, let me say that because you never know, man. You never know where you might go. You just never know. But I did some weather already, man, and for those zero, I understand. But boy, you gotta do what you gotta do. You gotta do what you gotta do. Hell yes. Sometimes you just gotta do what you gotta do. Damn. No, 4 hours. No, you mean tomorrow? Okay, 4 hours. No, like 4 hours. Oh, Fifi, that's bit. Hell yeah, boy, Blue. That's some serious goddamn weather. He walking into there, man. Zero? Damn. I know, man. That's no man. Damn, I know, man. I know. I know. I know I walk under 30, my goddamn toe them feel like I have no toes. My numb, my finger them like they want to fall off. I know, man. Okay. Ah, share. I won. Yeah, guys, man. Thank you guys as always, man. But we're gonna just try to keep it informational. This video really was not my video. It's a video I just run. I don't know if I might just take it down because it's really my video, or I might just take it down. But at the end of the day, we were able to learn something, and that's what's matter. You understand? We know about herbs, we heard about herbs, but sometimes to get the herbs from the plant, the process from taking it from the plant and carried into medicine. Able to learn that tonight or have an idea is a blessing. So shout out to that doctor for that information and the way he break it down. It was just amazing, man. Plenty information. I know it's a video that you might have to listen to a couple times to what I mean, because there's so much information to swallow or to deal with. but it worthwhile. 'Cause, man, you just never know. Because sometimes, because this is how things work. Sometimes somebody might have an idea of how to do something, and they share the idea, and then someone figure out another way, but at the same time you get in the same result. So by watching this, you could sit down, you know like me. I could put that, I will put that thing together. I'm telling you right now. Put it together, get the machine, put it together, and do that process just by watching. I'm telling you. And I'm sure there's many people who could do that as well. you know, or even though you ain't do it exactly how we doing it, you might figure out a other way. You know what I'm saying? But at the same time, you're able to get the medicine from the plant. It's interesting. It's really interesting. And as like I said, I enjoy how we break it down in the simplest form we can. It's a blessing. It's really a blessing. So yes, guys, we're gonna continue doing this like on a Friday night. I don't know. Sometimes I might do it on a Saturday, but most likely I'm gonna keep it going on a Friday night. But I'm trying to do it that we can learn something. We can at least learn something. Last week we did the herbs, and you see what I did. I showed a video last week with different herbs. Same people, same channel with different herbs. And now this week they show you how they extract the medicine from the herbs. Beautiful. I appreciate that. I appreciate them two videos last week and this week. I really do. Shout out to you guys, man. Let me see. Okay, guys. So I gotta say thank you guys, man. Thank you guys for coming out. I appreciate you guys again, man. No, tomorrow is another day. You guys may have work to do in the morning. My got garden work to clean up. Let me see. Okay, let me see. Took me back to chemistry class. One of my favorite. Oh wow, that's interesting, Audrey. That interesting. That's interesting. It if it take you back there, it gotta be interesting. And at the same time was one of your favorite class. Wow. I see why you into hobs like that, Audrey. I see. I see why you into hobs like that. That's wonderful. Wow. That's nice. That's nice. Really amazing. But don't want to keep you guys up, man. Just want to say thank you guys as always for coming out tonight again. Thank you guys. Truly appreciate each and every one of you for coming out. Thank you, Mike, Chaotic Garden. Thank you, Orgy Living Life. Thank you, Boy Blue. Thank you, Fifi Johnny, for coming through tonight. I appreciate you guys. And anyone else that come through, thank you guys so much. Have a blessed and wonderful weekend, man. All right, Boy Blue, good night, man. Thank you guys, man. See you guys soon. All right. Blessing to you all. All right. Yes, blessing everyone. Have a great weekend, each and every one. All right, guys. Peace and love. All right.